Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Tony Cox. Tony has spent more than 20 years at the Wellcome Sanger Institute prior to joining the UK Biocentre as CEO at the most opportunistic moment in January 2020. And little did he know that what lay ahead was the adventure of a lifetime. And he was awarded an OBE for his efforts to lead one of the lighthouse labs in the UK doing an enormous amount of COVID testing that we're going to talk about. But the UK Biocentre is also at the center of some of the most impactful population genomics programs of our time, including our Future Health. They were selected to be the partner for sample processing and analysis for this program that aims to genotype 5 million people in the UK. So we're going to spend some time talking today about the scale that some of these kind of programs and the speed that they're operating at today, but also where this might be going in the future. So Tony, thank you so much for taking the time and joining us today.
Oh, thanks for having me.
I'd love to go back to January 2020. What were you expecting going into the role? And then when did it all change and what happened from there?
Yeah, well, it was interesting because, I'd been thinking long and hard about moving out of the Sanger Institute where I was and what I was going to do. And this role came up and it was sort of, as advertised, it was going to be very much about doing some of the stuff that I was interested in, working at large scale in laboratories, doing sort of building big processes. And I was all fired up to come in and sort of learn about what the business was doing, how it was working. And I suppose I was only about a month, 6 weeks into the learning procedure, just really trying to get my feet under the table and understand how the place worked. And then all hell broke loose, as we all know. I remember very clearly in sort of the February, March, hearing about, the wave of infections that was happening out in the East and kind of watching it from the news reports track across the world. And it was really when it kind of got to, I suppose, Greece, and there was some those parts of Middle and Eastern Europe where the first cases, that's when I realized, oh my God, this is really going to happen. But at that point, it wasn't really, there was no obvious implication for us other than that, oh my goodness, this is going to be a big thing. So it wasn't until mid-March when I got a phone call from the government that it already kicked off.
And what did they ask in the phone call? Obviously, things evolved very rapidly from then, but what was the idea then? And then how did it evolve? I mean, the UK became probably one of the top couple countries in sequencing samples at large scale during the pandemic. So what was the ask originally then? And then how did it evolve?
Yeah, well, I think we're already, part of our history was connected with the National Institutes of Health Research and the Department of Health here in the UK anyway. So we were kind of probably on their sort of list of people to call.
Yeah.
So they phoned up and they said, well, we really have an important requirement. We want you to effectively drop everything and start testing COVID samples for us. And, at that stage, I was thinking, well, all of our projects that we are running are going to go to sleep anyway, because, nothing is going to be active during this. And we can either all go home and sort of wait it out, or we can step forward and sort of, take on the challenge and actually try and help out. So it was actually a very quick and very easy decision to make. I had a quick phone call with the chair of our board and say, look, I really think we need to do this because we need to do our bit and we can do something useful here. And from that point on, I got absolute sort of, solid support from everybody, not just above, but also, the team here at UK Biocentre.
And so over the course of the pandemic, how many samples did you end up testing and what did that ramp up look like? Did you have to make a huge amount of changes to the core infrastructure, or were you set up to run PCR assays, and you could kind of just ramp that to the degree required.
Yeah, I think, we had sort of fundamental experience in terms of running those types of assays. We had laboratories which were capable of holding, of sort of processing relatively large numbers of samples, thousands a day, for instance, but nothing on the scale that we were growing to. So, it wasn't oh my God, how are we going to do this? Because I was sort of at Sanger certainly used to building large processes, thinking about how you put together things at high throughput. That wasn't daunting in its own sense. It was more about understanding that we were delivering a diagnostic test, the quality of that process and the implications of the results were very important. So what we were doing had to be built properly, it had to run properly, and it had to be dependable. So there was a lot more, this wasn't just research,
Yes.
So I think there was that very definite aspect to building new processes. But we were testing samples, real samples, within a couple of weeks after that phone call. And we were given loads of help. There's help flooding in from every corner of the UK, both in terms of equipment and people to help us sort of get started, which was fantastic. I mean, it's easy to complain about the machinery and the bureaucracy of governments and sort of institutions, but I tell you what, when things all get behind a common purpose, it's really impressive what can be done, and nobody should sort of take that away from the effort that was sort of put in place. But the aspiration for the testing process grew just almost week on week. We were said, well, you need to be testing 1,000 samples, a week. And then suddenly it was 1,000 a day. And then there was, as the sort of the full impact, the realization of what the pandemic meant. We were talking to program managers from the government and from DHSC, and I was saying to them, look, you don't want to be thinking about terms of testing thousands of samples. You need to be planning for hundreds of thousands of samples in a relatively short period of time, because that's clearly what the implication is. Otherwise you're always playing catch up, you're never getting ahead. So yeah, that was really when we were tasked with going to 100,000 samples a day. And that's just a mad number of samples. Just what does 100,000 samples look like physically? If it fills rooms, they get delivered in articulated lorries. It's 24-hour operation, 7 day a week. It's where does the expertise come from? Where do all those instruments come from? So there's an awful lot of implications that sort of flood out from that initial sort of headline requirement. And I guess apart from just delivering 33 million tests over that sort of 2-year period, it was just a continuous cycle of build and then rebuild and scale and rebuild. So there was no time to stop and think about, how we're doing it and should we be doing it. It's just, let's get on with it, really. We were fortunate, I think, because Biocentre, we had space and configurable space, which allowed us to tear the whole building apart, in effect, and reconfigure it for one big sort of, monolithic workflow, and position sort of, resources and equipment around it. So it was an interesting time.
Yeah, to say the least. What are the biggest things that need to shift to go from 1,000 a day to 100,000 a day? What are the things that people you would expect or maybe wouldn't expect that start to break when you 100x like that?
Yeah, I think it's mostly it's about, space and scale and people. By the time that you're dealing with that, you need 50 people or 100 people unpacking samples.
Yeah.
Pulling things out of cardboard boxes, and then you say, okay, well, when you're dealing with 5 tons of cardboard a day, where does that go? If you've got seven tons of clinical waste a day, where does that go? We need 1,000 people to do it in shifts.
Wow.
How are we going to feed them? Have we got enough toilets? There are a whole series of sort of just questions that just flow out of having to deal with that. And you can either say, oh my God, this is impossible. You just knock over problems as they come up and you deal with them in practical or in completely impractical ways. One way or another, you just have to sort of think your way past the next problem. And that's really what it was about. There wasn't a sort of a master plan that was formed at the beginning, and this is how it's all going to unfold. It was very much a case of, Okay, so what are we going to be doing next week? What's the requirement for next week? And you just keep going like that. And it's a continuous process of solving small problems every day and keeping going.
Where does all that stuff go? Out of curiosity, how do you deal with tons of clinical waste and tons of cardboard showing up every day?
Well, I mean, we in the end we bought balers, and so we were palletizing bales of cardboard, and they were being shipped out daily to a recycling plant. Similarly, with the plastic, had to go off to clinical disposal sort of department company. But again, just sort of I mean, not difficult problems, but it's just about making sure that you think about how you're going to deal with things at that scale. It's underlying all of it. There's a requirement for quality. So, you
Yeah.
have to be working in an organized fashion. We work very closely with the NHS and with the accreditation authorities throughout, and they were fantastic partners alongside us. So we were getting advice. I think the one thing that I really sort of learned was that, you know, if people offer advice, listen,
Mm.
always, because people are incredibly generous with their time and their expertise. And it's a massive mistake to think that you know how to do it and you think that you've got the answers. You just suck in as much sort of advice and support that you possibly can. Because we had, with the army here setting up our warehouse, we had
Wow.
huge numbers of academic volunteers in the early months coming to help us set up laboratory processes. The Navy was delivering equipment. We had PCR machines arriving in the back of black taxis being sent up from London universities. I mean, it was nuts. But we had a great team from Deloitte who came in and helped set up our IT systems so that we could transfer results out really quickly to the sort of the distribution parts of the NHS. If you thought about it ahead of time, the complexity would just fry your brain. But just having all of those people here, getting them in, making them work together, just kind of giving them, fostering the environment where they can just do what they need to do is probably the most important part, rather than trying to kind of organize every little detail of how they want to work. It's
Yeah.
it's just you have to be kind of really flexible around it.
Do you think that one of the questions I've had around what we can learn and what we can't learn from the pandemic is you and a number of other people I've spoken to on the podcast have talked about what a catalyst that is for doing things that were just impossible before. You cut through the bureaucracy, you cut through this, planning paralysis. And we've seen this other times in the past in the genomics history, at least, like the Human Genome Project
Yeah,
was this way. But there was an urgency driven, I think, by international competition, right, between the private
absolutely.
sector
Yeah,
and the
yeah.
public. And we have a lot of moonshot projects that actually don't get that catalyst because maybe there's not a competitive element or a sense of urgency. Is there something to that, or what have you learned about the ingredients that actually allow you to cut through the noise in these situations?
Yeah, I think if you've got a critical mass of people who are really focused on the delivery, that everybody understands just how important. I mean, it helps if there is something really important riding on it. Just you could argue that sequencing the human genome was quite esoteric in one sense, but oh my goodness, can we imagine what would have happened if it wasn't sequenced in the way that it was? So
Yeah.
yeah, I think it's about having enough people who understand the implications of things going right, but also if things don't go right, the clarity with which everybody understood testing had to succeed, because you'll sit there and you think, well, if we get it wrong, the people might die because they don't know about stuff. And that's a really strong sort of motivating, driving sort of thread through everything that you do. And you don't cut corners, but you just don't worry about failure quite as much. You just, because I think we do spend a lot of time thinking about what will happen when or if things go wrong. And that, can paralyze you a bit. Nobody likes to fail. But I think if you've got that support around you and that there's the security of, well, if that didn't work, we can find another way to do it and that will be okay, then you're willing to take risks. And it's all about, your appetite to do something risky. Because, our testing could have all fallen over. We could have all just kind of screwed it up totally. But, there's absolutely no way that was going to stop us from trying. I think that's the important thing. It's just, taking that decision to put the first step forward, really.
Yeah. And so today you're doing fewer COVID related projects, maybe zero. I'm interested to hear
Yeah,
how that's
We're not.
changed.
We don't. There is no more COVID testing here. No.
Yeah. And so you've gone probably back to your roots of large scale genomics projects. Let's talk a little bit about Our Future Health. What other kinds of programs do you work on? Maybe you can give a flavor for the kinds of projects that you help drive.
Yeah, I mean, it's a broad range really. So everything from academic research projects, large cohort studies, through healthcare related big programs like Our Future Health, clinical trials. We've supported all the processing for the Grail Gallery trial,
Hmm.
one of these multi-cancer early diagnosis genomics tests. So a broad range of things. I mean, it's not all about just processing because, we have a very large biobanking and sample storage facility here, probably the largest certainly in the UK, millions and millions of samples. So for us, it's about trying to offer, not only the processing right the way, from currying samples around the country processing, but putting them into storage and long-term storage and biobanking. So it's a whole range of things. Some studies are, take advantage of that entire workflow, that whole sample journey. Some of them it's just storage, some of them it's just a bit of processing, but we flex into whatever's needed really.
Yeah, and are you doing sequencing and genotyping as well, or where do you end and where do some of the sequencing providers
We,
begin?
yeah, we don't do sequencing and genotyping ourselves. So what we do is we partner with commercial organizations who do that. Some of them actually on site here with
Mm.
us in Milton Keynes, so that we can offer a complete service, but without having to sort of, have all of that expertise in-house. I mean, I think, there's a lot of expertise required to do the things that we do at large scale, and it's another leap then to become, not only that, but also an expert in large scale sequencing or large scale data analysis. That's a big deal. So we at the moment, we just focus on that core activity of making sure that those samples end up in that genomic analysis in the best state that they can be.
Yeah. So I'd love to hear a little bit about the challenges that you may need to overcome with Our Future Health. So for those who aren't familiar with the program, it aims to recruit about 5 million volunteers across the UK. And it's going to be a cohort that reflects the population, I think, broadly with a range of ages, ethnicity, socioeconomic status. And there's a big focus on early detection and intervention around, diseases like Alzheimer's and cardiovascular disease, and using polygenic risk scores and other kinds of data to really I think make hopefully both basic science fundamental breakthroughs, but also translational impacts on the population. What's different and challenging about that project compared to some of the others you've worked on in the past?
Oh, well, I think the obvious thing is just scale. There are, thousands of samples coming into the organization every day, which, okay, on the scale of COVID, that's not a huge number, but the complexity of what we're doing with those samples is much greater, and therefore, it's more of a task. But when a blood sample comes in, it's often split into multiple aliquots. All of those aliquots are moving around, for different destinations. So it is a big sort of, tracking and sort of sample sort of movement operation inside Biocentre. Tens of thousands of samples, individuals, aliquot samples every day, going through, getting prepped for analysis, going off to partners for genomics, getting stored, getting retrieved. There's a lot of complexity in moving that around and doing it, so that, everything is absolutely precise and traceable. So, I think the real challenges are scale, but also there are some fairly stringent requirements on processing time. So some of these samples have to come in, they have to be processed within a fixed window so that the maximum amount of integrity is built into that sample for the long term. So there's some pretty stringent sort of requirements for how that processing is delivered, which keeps us, as you can imagine, very busy.
Yeah. What's the footprint of the facility? Like, how big are we talking to do these kinds of projects?
Couple of big warehouses, I suppose, if you
Yeah.
were to think about it like that, joined together. I think the one benefit of the organization in terms of just the overall facility, it used to be the old Hewlett Packard check processing facility before it was converted into Biocentre. The one benefit of that is it has a really big and powerful kind of power supply here. So, a lot of the equipment that we have put in, we've not had to do significant alterations to the infrastructure, because we run half a dozen very large automated freezer systems, quite power hungry. You couldn't drop those into an ordinary industrial unit. So
Right.
I think the people who kind of identified this building and thought, yeah, that's the one for Biocentre, really did a good job.
Yeah, and I was imagining there's also probably an important security element, not necessarily from like people breaking and entering, but from the elements, right? You've got these precious samples that you're biobanking, and they can't get flooded. They can't, the freezers go down. You got to have backup. What does that whole operation look like to make sure that you're not only keeping them for decades in some cases, but you're providing at least as many guarantees as you can that they're not going to get wiped out by a natural disaster.
Absolutely. Yeah, no, I mean, that's critical to us, critical to many of, our collaborators and customers. So, I mean, there are two ways that we approach that. One is that we can split collections and we can collect at least a proportion or an aliquot of the collection at our satellite site just outside Oxford. So, we physically share the risk. But on site, we have obviously, backup power systems, both battery and then generator backed. If things like, the generators failed, then we have the option to cool the freezers with liquid nitrogen. So
Right.
there are multiple kind of layers of disaster recovery on that. So yeah, I mean, if those things run out, then I think we've got bigger problems.
Yeah, that's right. Yeah.
But yeah, I mean, we really do focus and make sure that those systems are really solid, because like you say, some of these collections have been here for 10 years and will be here for probably decades more. So we're very aware of the importance of what that integrity means for the utility of the sample for the long term.
Yeah. What is the most sample intensive thing that's potentially on the horizon for us? I'm imagining another pandemic is about as gnarly as it gets, doing hundreds of thousands of days. But can some of the serial sampling for cancer screenings and these kind of things, if they went population wide, is that also a similar scale where you could be doing tens of thousands of samples a day? Or what do you think about the things that push the
Yeah,
envelope on this?
I think we're at an interesting point now, certainly in terms of where genomics has certainly penetrated to some degree clinical care. I think it's over the last few years, it's begun to prove its value and its utility. And I think, there are a lot of people now thinking very carefully about, what are the wider opportunities? Should it become a standard in terms of a clinical tool? What are the implications of that? I don't think there's any doubt that there is huge clinical utility there. It's about how you deploy it, how you scale it up, how you make it, time relevant, and lots of questions around that. But I think the underlying sort of imperative is pretty clear.
Yeah. Yeah. What are you seeing the most potential on there? Is it things like polygenic risk scores? Is it the minimally invasive kind of cancer testing like Galleri that you mentioned, or others?
Yeah, I suppose in terms of impact, probably cancer testing, and clearly it's been deployed extensively in that regard. I think it's the challenge is how you scale that up and how you move it away from purely diagnostic to almost a surveillance setting, so that you can take advantage of the tool for earlier diagnosis. I think, obviously there are a lot of studies out there in the wild now in the UK and elsewhere looking at the potential and the benefit of doing those sorts of studies. Obviously in the UK, everything is likely to be delivered through the National Health Service, and they in their own right are sort of looking at those. But that, cancer, I think the burden of rare disease across the population is high, not necessarily appreciated by everybody outside the sort of the research and genomics arena. So I think the utility of using those to help people who might have rare disease, but reduce the cumulative burden and that very expensive burden on the healthcare system. So I think there are loads of opportunities. Taking it into general health monitoring, I think, is a really interesting, interesting area. I think OFH is definitely sort of forging into that world, proving that with the right information, the right metrics, the ability to give all individuals data which allows them to make sensible choices about their health risks is vitally important. Moving the NHS from arguably an unaffordable mode of treating people when they get sick to one where we help to prevent people from getting sick, or certainly until much later on in their lives. So I think the potential there is absolutely enormous. I think it's about how we build and deploy systems that are robust, reliable, that are trusted, and how they can really build out a system for the benefit of all.
Yeah, absolutely. I think the whole kind of preventive health piece feels like it's picking up a lot of steam, not just on the genetic side, but also we had a great podcast guest, Austin Argentieri, talking about a proteomics based measure of aging that they've developed and some of the insights there. And it feels like we could live in a world in a decade where you have a lot more frequent sampling and a lot more intelligent outputs in terms of what that actually means for your health from some of these really broadly applicable tests like proteomics and genomics. But the clinical utility point you made is key to it all, right? We have to figure out how to pay for it and how to make it work and how to do it at a scale that makes sense.
Yeah, and I think there's obviously a lot of work needed to examine the health economics of this, and I think we've in a way we've got to have faith and invest in a system that will begin to pay back in 2 decades or 3 decades, and have the sort of be bold enough to take those steps and believe that that is the right thing to do.
Yeah, and is one of the things, and I don't know if this is true actually. If you know anything about this, I'm interested to hear it from a more authoritative source. One of the things that I've heard is that when it was discovered how to create iPSCs, stem cells, that depending on how a sample was stored has a dramatic impact on whether or not you can actually successfully do that. And it also makes me think about the things that we don't know about sample storage right now. There may be a technology that comes through in a few years that makes us either be thankful that we stored our samples in a certain way because they enabled us to do that across millions, or say, gosh, I wish I'd known that we needed to store it this way. Is that the case with the impact of sample storage on some of those you're almost future proofing yourself if you do it in the right way, right? I wonder if you could talk a little bit about that.
Yeah, I think you're absolutely right. I think if you can store a sample with maintaining as much integrity as possible, you effectively keep all of your options open, or as many options open as you can think of. Yeah. I remember some arguments made a few years ago about, the advent of sequencing, high throughput sequencing. There's no point in storing a sample because it's much easier just to re-extract something and sort of, rather than the cost. I think, we've gone full circle on that. And I think, the long-term storage of samples, not only for its genetic, but also for its proteomic and metabolomic sort of opportunities, is becoming clear as those new technologies come on. I think we understand now that there's a lot more information to mine out of a seemingly sort of fairly simple sample. And those things can have, enormous information value, both for research, but also for healthcare. So, yeah, I think the idea of storing samples for the benefit of having those samples in time, not just because they're regulatory requirements for clinical trials or something. I think there is a more general understanding of that. Yeah.
Yeah, our future health or any other projects that you work with thinking about really long-term serial sampling. One of the big gaps that I often hear about in, biobanks and population genomics programs is we may have a sample from a moment in time or 2 moments in time, but to have every year for decades, for example, we don't know what could be discovered by having that in some of the chronic diseases that we have, to just really understand the journey from pre-symptomatic to symptomatic, or to perisymptomatic to symptomatic in a disease like Alzheimer's or others. Are there people who are working on that or thinking about that?
I think so. I mean, I think things like the UK Biobank and some of the data that's coming out is definitely prompting those kinds of thoughts. I mean, I think having the right consent structure in a project that allows you to recontact, participants and ask them, and also, set the scene early on in the study where you can create the expectation that, we would like to come back to you, once every 6 months for some blood or whatever. I think there's a general understanding that those things are going to be much more important or much more interesting or have much more value. So, yeah, I think, it's obviously much more expensive and much more elaborate to run those sorts of studies. getting funding for those kind of studies that will last years and decades is much more difficult.
Yeah.
And I suppose that's why IFH and its aspiration for generational sort of studies, I think, is really exciting. So the promise is very high.
Yeah, I couldn't agree more. One question I wanted to ask you to close out that I ask a lot of our guests is if there's any technology project, something that you're really excited about that you find really interesting that may not be on everyone's radar yet. So a few years ago, people were talking about, Olink proteomics, and it's going to be the next cool thing that is going to happen in the UK Biobank. What are you thinking about right now that is really interesting that maybe isn't on everybody's radar?
I don't know whether about techniques, but I mean, one thing that struck me, I've been at a couple of meetings just recently in the last few months, and one of them was, a rare disease meeting. And one of the things that struck me was the potential for antisense oligonucleotides, ASOs for treatment in rare disease. And the thing that I found really fascinating and really exciting was, how do you solve that problem where you create a therapeutic for not just a rare disease, but for an individual with a rare disease, where, creating a therapeutic that is absolutely tailored to just one person and will never be, used for anybody else.
Yes.
I mean, I think a few years ago, the kind of how on earth would we ever approach doing that was, it was still a mystery. But there's a lot of interest in doing that now. There's capabilities around ASOs for doing that where it's a relevant therapy, but also there's engagement with the authorities who are sort of willing to understand what the regulatory kind of difficulties are and to support those approaches. I just thought the potential for that and the fact that we could approach that level of personalization for therapies really exciting.
Yeah.
That blew me away.
That is a great one. I didn't, for everyone listening, I did not prep Tony on this question. It came as a complete curveball, but I think this is a brilliant answer to it. And there's actually a company that, if they're not on your radar already, they should be. It's called Everyone Medicines. They're based in the US. And I think their goal is to try to do this at scale. And they're co-founded by a mother of a child who went through one of these N of 1 therapies in the very first time and had this experience both building a nonprofit and with her family, and has tried to turn this into something that could scale and build up. I should reach out to them and see if we can get someone from Everyone on the program. But I think this is a really exciting direction. I couldn't agree more with you. Well, Tony, thank you. This was really great. If people want to keep track of you, keep track of the UK Biocentre, I'm sure your website is a great place for it. Is there anywhere else that you tweeting, or are you doing anything?
Yeah, we're present on Twitter, and be very happy to get in touch on LinkedIn, where we post and sort of try and keep people up to date with what we're doing. So yeah, it's been fun.
Wonderful. And that's ukbiocentre.com, center spelled the UK way, CENTRE. Right. Thank you again, Tony. I really appreciate it.
Thank you very much. Much appreciated. Cheers.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.